During the early days of nuclear physics, scientists faced a serious moral dilemma due to the possible uses of atomic energy. They knew what enormous potential it had in providing energy for mankind, but they were also clear about its destructive power. The explosive development of genetics has raised similar ethical concerns, so the scientists themselves asked their governments to set up a strict system of regulation. This film demonstrates the opportunities presented by suitably regulated gene technologies in overcoming the most urgent problems facing mankind. József Pálinkás, atomic physicist, president of the Hungarian Academy of Sciences. Over 1 million species are to be found on the Earth. The design and characteristics of all the individuals in this exceptionally rich living world are determined by the simple coding system of the hereditary material present in the cells. The 4 main components of the DNA are the same in all species, but their quantity and order are diverse. Nature is thus a master of composition, creating everything from the most primitive protozoan to the vertebrates at the top of the evolutionary ladder through the variations on these 4 basic themes stored in the genetic code. Although genetics is still a young science, the structure of the hereditary material has only recently been deciphered. The process of heredity and the benefits arising from its manipulation were already known in the ancient world.
A különböző népek vándorlásaik során
migrating people obtained food by collecting, fishing and hunting. Later, when they settled down, they grew or kept the same plants and animals. Efforts to retain and multiply favourable characteristics were the first steps in breeding. Agriculture led to advanced civilizations in Mesopotamia and the Nile Delta of Egypt, and to the Mayan and Incan cultures.
A nemesítésnek a kezdetét jelentette. Ahol ilyen alapokon indult meg a mezőgazdaság, ott fejlett civilizációk alakultak ki, gondoljunk csak Mezopotámiára, vagy pedig Egyiptomra, a Nílus-deltára, illetve a maja és inka kultúrákra.
A buza nemesítése természetes úton lényegesen hamarabb kezdődött, mint az ember által végzett tudatos nemesítés.
The natural breeding of wheat began far earlier than the first human efforts. The wheat we know today developed from crosses between 3 different wild grass species, which is why it has such good adaptability and can be grown in almost every part of the world.
Nagyon jó alkalmazkodó képességgel rendelkezik.
Európa mezőgazdasága
Relatively few species were originally cultivated in Europe. After the discovery of America, numerous crops such as maize, potatoes, tomato, and paprika were introduced into Europe. Under the new favourable climatic conditions, these species soon became established. Nowadays, more than 95% of the crops cultivated in Europe originate from other continents.
Napjaink mezőgazdasága Európában több mint 95%-ban olyan növényeket termeszt, amely más kontinensről származik.
The biological multiplicity described by Darwin is now preserved in gene banks, which are the pledge of long-term stability in quality crop production and animal husbandry. There is a cereal gene bank in Martonvásár, for example, while indigenous variations of poultry are bred in research institutes for animal husbandry and feeding in Gurulu.
A génbankban fenntartott őshonos magyar baromfifajtákat, tyúkfajtákat
When indigenous varieties of poultry from the gene bank were compared with samples from other variety collections and with commercial stocks, Hungarian varieties formed a separate group, clearly distinct from both commercial varieties and other indigenous European varieties. This shows that they have great genetic potential, which would be well worth exploiting by Hungarian breeders.
Őshonos fajtákhoz képest, és ez is rámutat genetikai potenciáljukra, értékükre, amelyet nagyon érdekes lenne és értékes lenne hasznosítani a magyar tenyésztés számára is.
The basic laws of heredity were first discovered in the mid-19th century by an Augustinian monk from Moravia, Gregor Mendel, who had no knowledge of DNA or of the DNA sections coding specific characters known as genes. In experiments on peas, he crossed male and female lines with easily distinguishable traits, for instance round or angular seeds, yellow or green seeds. He then crossed the members of his first hybrid generation among themselves and noted the appearance of the parental traits in each generation, making a statistical evaluation of their frequency. He considered what he called a regulatory factor to be responsible for the regularities he noted.
Az élet titkának megfejtésében mérföldkövet jelentett Watson és Crick felfedezése, a DNS szerkezetének meghatározása.
Watson and Crick's discovery of the structure of DNA was a milestone that led to a revolution in molecular biology. It became possible to determine the primary structure of the hereditary material, to identify sections coding for various traits, and to isolate them from one organism and transfer them to another, resulting in genetically modified organisms, or GMOs. The prejudice against GMOs is due to the lack of proper information. Most people have no idea what GMOs really are.
Az ellenérzés a társadalomban a GMO-kkal szemben az információ hiányra vezethető vissza. Az emberek többsége nem tudja, mi is az a GMO.
Genetic modification is a method by which DNA segments responsible for new traits are introduced artificially into living cells. During cell multiplication, the gene coding for the desired traits is inherited by the progeny cells. The use of genetic modification in stem cell research allows various diseases to be modeled.
A valódi embrionális őssejtek azok a szervezet összes sejttípusát képesek létrehozni.
Embryonic stem cells can produce all cell types found in the organism, including skeletal muscles, heart muscles, tissue and skin cells, liver cells, blood-forming cells, and nerve cells. Embryonic stem cells from mice can be used to develop cell lines in which the gene coding of the desired trait can be modified. The transgenic animals derived from these cell lines can then be used to study the effects of the genetic modification both during embryonic development and in adult mice.
Több mint 6000 ilyen transzgénikus egeret sikerült létrehozni.
More than 6,000 transgenic mice have been created, including animals with developmental deficiencies, allowing problems related to vein formation, blood formation, heart and muscle development to be modeled. Not only diseases were modeled, but also the functioning of genes involved in early embryonic development.
Modellállatokat lehetett létrehozni, hanem a különböző a korai embrionális fejlődésben szerepet játszó gének működését is meg lehetett ismerni ezeken az állatokon keresztül.
Embryonic stem cells are easy to culture under laboratory conditions. Within a few weeks, colonies of several million cells can be produced from 100 initial cells. The genetic modification of the colonies is achieved via DNA electroporation, a technique by which DNA can be introduced into millions of cells. Scientists then select viable cells in which the gene has been incorporated into the desired locus and is capable of functioning. On average, 10 cell colonies with the desired genetic modification are detected for every 10 million treated cells. If the gene construct used for this technique contains regulatory components, the scientist can target a specific tissue or organ for gene expression and can determine whether the gene should begin functioning automatically or only in response to an external stimulus. Animals genetically modified to produce useful compounds, and thus known as bioreactors, are being used to find cures for numerous illnesses. Proteins that play an important role in the defense system in the human body can be produced in the milk of various animals.
Az általunk előállított génmódosított állatok.
In these genetically modified animals, antibodies are produced more rapidly and in larger quantities than in normal animals, but in equally good quality, and so fewer animals are required. This means that plenty of antibodies are available for diagnosing tumors.
Állatra van szükség, és a különös jelentősége az, hogy ezeket az anyagokat nagy mennyiségben fel lehet használni a tumordiagnosztikában.
Sokan úgy ítélik meg,
Many people fear that genetic modification could have unforeseen consequences if we change characteristics that have been fixed once and for all.
változtatunk meg.
But the living world is in a state of constant development, and evolution is by no means finished.
És az evolúció egy végeláthatatlan folyamat.
Breeders hoping to ensure safe food supplies for mankind by modifying the genetic material of cultivated plants also utilize this technique to change the traits that are important for the production technology. It's as if the time required for evolution was being shortened. The plant genome is extremely sensitive. The activity of various gene sections may fluctuate wildly in response to changes in environmental factors, such as light intensity, rainfall quantities, or temperature. If gene function is so sensitive even to external conditions, what effect is the introduction of a new gene likely to have on the behavior of the genetic material? Will it induce unexpected, indirect, undetectable changes that could be a danger to human health or to the environment? The DNA chip technology is designed to give exact answers to these questions.
Természetesen az a DNS chip nem csak arra alkalmas,
This
hogy
DNA chip can be used to compare gene functioning in various environmental situations, or in plants with different gene pools. For instance, 2 different wheat varieties, 2 different rice or apple varieties, or varieties of other plant species. The functioning of normal and gene-modified versions of plant variety can be compared to discover what changes are caused when we add a new gene to the tens of thousands of genes already present. The results indicate that the addition of a new single gene may alter the functioning of a handful of genes, but these changes are usually negligible compared to the changes caused by a slight change in temperature, for example. Environmental conditions, soil, cultivation technology, or spraying with chemicals lead to far greater alterations in the gene expression pattern than the introduction of a transgene.
15-20 vagy 35-40 ezer génje mellé.
It is predicted that global warming will have an intense effect in Hungary due to the special nature of the Carpathian Basin. Any project that aims to help crops to adapt to the changes in the climate is thus of vital importance. The team led by Zsófia Bánfalvi in the Gödöllő Research Institute is working on the development of drought-tolerant potatoes. The high water demand of this species means that in dry years it can only be grown profitably if it's regularly irrigated. Scientists are looking for genes that could help the plants to produce a satisfactory yield, even if the rainfall supplies are poor.
A burgonya szárazságtűrő képességének javítására
A gene from baker's yeast was first used to improve the drought tolerance of potato. Yeast was an obvious choice, as it had a good tolerance of drying out and revives quickly when remoistened.
a szárazságot, jelentékeny vízveszteség után is újraélet, hogyha nedvesség éri.
But how does the gene get into the potatoes? A plant-infecting bacterium exists, which is capable of taking up various DNA segments and then introducing them into target organisms. Scientists first incorporate the desired gene into the genetic material of the bacterium, after which they make tiny cuts on the potato leaves and infect them with the bacterium carrying the gene. Once the gene transfer has taken place, the bacterium is killed with antibiotics, and leaves containing genetically modified cells are placed on shoot-inducing medium. Once shoots appear, they are rooted to produce new, genetically modified potato plants. The seedlings are then tested in the greenhouse for drought tolerance and yield.
A sütőélesztő génjében módosított burgonyák
Potato plants modified with the yeast gene were more drought tolerant and stayed green, even with lower water supplies, but the tuber yield was only half that of the original variety, so a new solution was needed. It proved unnecessary to use alien genes, because drought tolerance can also be enhanced by inhibiting the expression of certain genes present in potato
vannak olyan
plants.
burgonyagének, amelyeket ha nek a működését gátoljuk, akkor ezzel is fokozni tudjuk a szárazságtűrést.
Using this special form of genetic modification, the research team aims to develop varieties capable of producing yields approaching those of conventional plants, even with minimal rainfall supplies. Potato plants raised in the greenhouse are then tested in field experiments in Keszthely.
A burgonya esetében is, mint ugyanúgy minden más genetikai módosított növényfaj.
For all GM plants, there are questions of food safety and environmental safety. In the case of potatoes, the question is what effect the release of GM plants in the field will have on the human environment. This depends partly on the gene or gene construct, but also on the type of crop. The risk is far greater for open flowering crops like alfalfa or wheat than for potato, which has no wild relatives in Europe, so there's practically no likelihood that the modified gene will escape by outcrossing with other species.
Nézzük a kérdést, hogy a burgonya, mint ilyen, lényegesen különbözik-e mondjuk egy szabad elvirágzású növénytől, mint amilyen egy lucerna, vagy egy öntermékenyülő növény, de szintén szabad elvirágzású, mint a búza, akkor azt kell mondani, hogy lényegesen kisebb környezeti kockázattal járhat bármilyen gén beépítése és annak a környezeti kibocsátása burgonya esetében, hiszen Európában nincsenek olyan vad rokonfajai, például a burgonyák, amellyel szabadon kereszteződne, így egy gén megszökés esélye gyakorlatilag nulla.
This indirect form of gene transformation, with the help of a soil-borne bacterium, is simple to apply in potato and in most other dicotyledonous plants. But monocots, such as cereals, are not host plants for this bacterium, so special techniques are required if it is to be used to deliver DNA. Scientists in Gudelu thus used a direct ballistic technique involving a computer-controlled gene gun.
A génbelövéses eljárás lényege az, hogy a bejutatni kívánt
The technique consists of attaching DNA molecules to 1 micrometer gold or tungsten particles and shooting them into the plant tissue using a gene gun.
A sörétes puska elvén az élő növényi szövetbe belőjük.
The particles used to deliver the genetic material penetrate the cell wall and enter the cell. As a large number of particles are used for bombardment, there is a high statistical likelihood that some cells will survive the treatment and the desired DNA segment will be incorporated into the genetic material. Gene bombardment is also utilized in the fight against fungal diseases in wheat. Rust fungi are pathogens that infect the leaves and stems of wheat, causing yield losses of as much as 30 to 50%. Protecting the crop by spraying damages the environment and is also extremely expensive. Certain segments of the genetic material make wheat resistant to disease. Breeders have attempted to improve this capability, but have been unable to develop complete protection because the activity of the genes responsible for rust resistance is too weak. Plants produce fungicidal enzymes, but not enough and not quickly enough. The team led by Barnabas Jenes cooperated with the fungus research team to find a gene that could be used to enhance wheat disease resistance. A DNA segment coding for the desired trait was found in a fungus species that lives as a parasite on other fungi and has long been used in biological plant protection. The gene responsible for enzyme production in the pathogen-destroying fungus can be introduced ballistically into wheat tissue cultures in order to develop plants tolerant to rust fungus.
A beépített transzgén a búzának csak a zöld részeiben, tehát
The gene is only active in the stems and leaves of the wheat plants, where it provides protection against stem and leaf rust. The gene is not active in the grain, so that the enzyme it produces is not present in the flour.
ezért az általa termeltetett enzim nem fog megjelenni a lisztben sem.
In addition to pathogen attacks, plants must also cope with environmental stress. The development of tolerance works on the same principle as resistance to rust fungi. The genetic program is partially rewritten to introduce the overproduction of detoxifying protein, which is a major component in natural defense mechanisms.
A genetikai transformáció olyan lehetőséget rejt a...
Genetic transformation allows wheat breeders to develop a new variety in a single step by introducing a gene to improve resistance to stress, insects or fungal disease, or to enhance the technological quality. There is nothing really new about this, because genes or chromosome segments from alien species have been introduced into wheat before. A rye chromosome segment containing several extremely useful genes was introduced into wheat in the early 20th century and gradually spread all over the world, leading to much greater adaptability and better yield potential and stress resistance. Wheat containing this rye segment are now grown on millions of hectares every year and have been used by Martin-Vachar wheat breeders to develop new varieties. Everyone can see that this has caused no environmental damage and has contributed to enhance yields and improve stress resistance.
Látható, hogy a nem transgénikus...
These two plants were both exposed to the same level of drought stress, a quarter of the normal water supply. The plant without the transgene is very short, with very small ear containing only a few grains.
És néhány szem található csak a kalászban. A másik növény, amely ugyanilyen...
The other plant, grown under exactly the same conditions, with only a quarter of the normal water supply, is taller, with twice as big an ear and a lot more seeds, because the transgene helps the plant to survive drought stress.
Azért a stressz alatt egy szebb produkciót mutatott, hosszabb a növény, a kalásza is duplája, mint az előzőnek, és a szemtermés is lényegesen több benne.
Another important crop is maize, the production of which will also have to be reconsidered in the future due to the appearance of the corn rootworm. The first specimens of this species, now one of the most serious plant protection challenges in Hungary and neighbouring countries, were observed in southern Hungary in 1995, and by 2003 the whole of the maize-growing area of the country was infected. So farmers have spent around 4 billion forints a year on fighting this pest, which still cause yield losses amounting to 20 to 25 billion forints each summer. Conventional means of control, crop rotation, soil disinfection and spraying, are now unable to provide complete protection alone. So scientists are attempting to develop resistant hybrids.
Martonvásáron a mezőgazdasági kutatóintézetben
Two techniques are used in Martonvásár to develop tolerant or resistant hybrids. Conventional methods are used to improve the tolerance level so that the hybrids have smaller yield losses and lodge less when attacked by corn rootworm. The other technique involves using a gene from the bacterium Bacillus thuringiensis into the plant.
Melynek során egy baktériumból, a Bacillus thuringiensisből kivont gén beépítésével
This produces a toxin that is specific to the corn rootworm. The toxin not only protects the plant from larval attacks, but also destroys the larva, thus reducing the insect population. Concerns have been raised about whether this toxin might be dangerous to other organisms, but its action spectrum is much narrower than within the chemicals used for soil disinfection, which kill a wide range of insects, while this toxin only damages bugs and has no effect on other insects.
A biztonsággal kapcsolatosan fölmerülő kérdések, miszerint ez a toxin vajon veszélyes más nem célzott szervezetekre, mondhatjuk, hogy a hatásspektruma lényegesen szűkebb, mint egy kémiai szernek a hatásspektruma.
Tehát mi is az a GMO? Lehetséges válasz.
So what are GMOs? They're a possible answer to global problems, including the economical production of basic foodstuffs, the preservation of biological diversity, environment protection and human medicine. However, any technology will only serve the interests of mankind if it's adequately supervised.
Körülmények között alkalmazzuk.
In 1971, the American geneticists who developed the technology approached their government to ask for a moratorium on experimentation while possible risks were evaluated. Since then, research all over the world, including the European Union, has been strictly regulated. The Julius Kuhn Institute in Quedlinburg analyzes the risks raised by the new technology and investigates how it can be applied safely.
We are working, for example, on tools for precision biotechnology to lower the regulatory burden before placing on the market, for example, marker gene elimination from transgenic plants. We are also working on biosafety aspects of molecular farming, so when you want to produce pharmaceuticals in genetically modified plants. We are also working on methods to reduce the gene flow for special applications, so that the GM plants are not producing pollen or the flowers are closed and the pollen cannot be provided to other plants.
Általános a félelem attól is,
There's a general concern that once a plant has been introduced to the environment, it can no longer be removed. A good counter-argument is the futile attempt to introduce cotton production in Hungary in the 1950s. Not a single cotton plant or cotton gene can now be found in the Hungarian flora.
Mezőgazdaságunk ebben az évben több mint 10 000 holdon termel gyapotot. A mezőberényi állami gazdaság területén szeptemberben kezdődik a gyapotszedés. A Szovjetunióból meghonosított gyapot nálunk alacsonyabb szárú. Ezzel biztosítható, hogy a mi éghajlatunk alatt is beérik.
Behind me is a field experiment within a national program in Germany, and here we are working on the topic biological mitigation techniques, and the idea is to use biological mitigation techniques in order to reduce the potential of pollen-mediated gene flow, to use cytoplasmic maize, sterile maize, as a containment strategy, because if the varieties are really stable, they are not able to produce any pollen. Due to the fact that we are working on pollen-mediated gene flow, we need to separate our own field experiments from other field experiments, because from neighboring fields the pollen can flow into our field, and we don't want that. That's why we cultivate hemp. Hemp grows higher than maize, and that's why the pollen cannot go into the maize production field.
For more than 20 years, the International Centre for Genetic Engineering and Biotechnology has organised seminars on the environmental and health aspects of genetic modification for those working on the regulation and application of gene technologies. The meetings organised by the ICGB in the small Italian village of Cotron are chiefly attended by participants from developing countries.
They are coming from at least 25 different countries, which span from Cameroon, Nigeria, Kenya, Latin America, Costa Rica, Argentina, Brazil, India, South Africa. Most of them are from developing countries and Central Eastern European countries as well. We have developed a number of talks and presentations which provide the students and the participants with the various aspects that the decision-making process needs to look at from the point of view of the assessment of any possible risk connected or related to the use of genetically modified organisms in agriculture.
GM is a technology that is being already for 15 years. Some people say it's the upcoming technology. We say it's the now technology. It's not something that it is upcoming, it's right now. We cannot negate that they are already, it's a technology already growing, and it's an upcoming movement that is not going to be easily stopped. So we have to see how we can adapt it and use it for our own benefit without damaging the environment or human health.
I believe that the GMO is going to be a case. Already we have GMO BT cotton already approved in India, and the regulations are in place, and I think India is going to play a major role in GMO research as well as GMO applications. GMOs will only come through the traditional plant breeding. It has to go through the same process again. Whenever the new GMO or an event comes into the table, and specific protocols are applied, and they are going to be assessing. So mostly it is a very well established, I would say, biosafety guidelines in India, and quite in fact much tougher than any of the regulations you can see in the world. So that point of view, there will be a very strict vigilance before release and as well as after the release.
Az európai társadalmak aggodalmai.
Due to public concern in Europe, the EU set up a food safety authority in Parma, Italy, which is staffed by experts as thoroughly trained as those in the US Environmental Protection Agency. The difference is that people and the government of America trust the recommendations of their experts, while the EU politicians query the competence of the authority they themselves set up.
EFSA's main role in GMO risk assessment is to provide advice to the risk managers, that is the European community and the member states, on the safety of GMOs with regard to the human, the animal health and the environment. So this is a very global mission statement of EFSA. To do this, we have a panel of 21 experts in the GMO area with a very broad range of expertises. So we have people from the molecular sciences, breeding sciences, toxicologists, nutritionists, until the side of the environmental risk assessment, ecology, entomology, weed sciences. And this group of people are, in addition, helped by about 40 external experts. They organize themselves in working groups, and all this expertise together assesses then applications that we receive. All our scientists are selected based on their proven scientific excellence. And from the independent point of view, we have a very stringent system. The first step is the so-called completeness check, and this is something for the EFSA staff scientists. We have a unit, a GMO unit with 17 scientists. So they take a first look at this dossier, and then they decide whether or not the dossier complies with the standards with regard to completeness. We call it completeness check. And if that is found to be complete, then it's made valid. And this is the time point where it actually enters the risk assessment phase. So then we distribute this dossier to our experts. The decision in the end on how a scientific opinion looks, on the text of a scientific opinion, is really reserved for the GMO panel.
The world population is growing by 70 to 80 million a year. Around 40,000 hectares of land is required to house 1 million people. So each year, the amount of land available for agriculture is declining by 3 million hectares. In addition to urbanization, the area of arable land is also being reduced by desertification and increasingly extreme weather conditions caused by global warming. So the food supplies of mankind can only be ensured in the long term by increasing the yield per hectare. This goal could be achieved using GM plants with enhanced tolerance of drought, pests and pathogens, allowing them to be grown more cheaply and efficiently, with less damage to the environment, and achieving yields equal to or surpassing that of conventional varieties, as they require less water and the use of chemicals can be limited or reduced to a minimum. In 2008, GM crops were grown on 125 million hectares, almost 10% of the global arable area. This may rise to 15% by 2015, as the sowing area has been increased by 15% a year. GM crops are grown in 25 countries. A further 30 have authorized the import of GM plants. GM crops are grown on 80% of the total soya area and 30% of the total maize area. In major exporting countries, the USA, Argentina and Brazil, this proportion is 90%. These three countries control 90% of world trade. A very high surcharge of 50 to 80 euros is now charged for non-GM soya and maize, compared with a purchase price of around 100 euros for maize and 300 euros for soya. Neither consumers nor feed merchants are prepared to pay this.
Élelmiszer, az import és az élelmiszer takarmány célú felhasználás megy.
The volume of GM food imports and use for feed purposes is high in Europe, but very little is produced, which is absurd. GM maize and soya pours into Europe and is eaten by humans and animals. This means we acknowledge there is no health risk, yet our own farmers are not allowed to grow it. This is nonsense and completely incomprehensible.
Ugyanazt a kukoricás szóját a saját parasztjainknak meg nem engedjük, hogy termesszék.
A genetikai módosított növények termesztésének az lenne a lényege.
Despite the mistrust of politicians, we need to grow GM crops to reduce external risks and make farming incomes more predictable. This has been a major problem recently, mainly because of external factors such as climate effects and the appearance of new insect pests. These have had a negative effect on profitability. At present, we grow GM MON 810 maize on 1,500 hectares. This is BT maize, which is resistant to European corn borer. It is grown partly because it yields 8% more than the non-GM variant of the hybrid, but more importantly, it's healthier than conventional maize. As the plants are not chewed by the pest, the plant tissues are not exposed to fusarium fungi that produce toxins. These are extremely poisonous to both humans and animals. One toxin, aflatoxin, may kill pigs or poultry that eat contaminated feed and certainly stunts their growth. This is the main reason why livestock farmers prefer this type of maize as feed.
Favorizeze apariția fuzariozei de regulă și, sigur, după aceasta a alfa toxinelor care sunt atât de dăunătoare pentru viață.
Most sem fog sikerülni rákényszeríteni azt, amit nem akarunk.
A két évtermés különbsége nagyobb ennél, az ígért előnynél. Helyén valóbb lenne több állatot tartani, és nem génmódosított takarmánnyal ellátni őket.
Amiben nem vagyunk biztosak,
hogy környezeti és humán egészségügyi kár nem fog okozni.
Ez politika, tehát vannak a nagyon elutasító negatív tagállamok, Magyarország.
This is simply politics. Member states like Hungary, Austria, Greece, France and Luxembourg have a negative attitude and say Europe does not need this technology. They manufacture so-called proof of environmental risk, for example the risk of moths dying, and submit them to the European Food Safety Authority, which is maintained by EU taxpayers. When the EFSA refuses to accept the evidence and pronounces the product safe, these countries call for a moratorium based on their own laws and often influence the more positively thinking states. So these EU states are not keeping to the EU laws.
A célra fenntartott testület. Ezért tartjuk ezt a tudományos tanácsadó testületet, hogy ezekben döntsön. Majd miután ez a testület azt mondja, hogy nem állják meg a helyüket ezek az eredmények, amiket a negatív tagállamok benyújtanak, de hiába mondják ki, hogy ez ne, és a termékbiztonságos, ezt is kimondja az EFSA, hiába adott tagállam simán a saját eredményére hivatkozva elrendeli a moratóriumot, és rendszerint befolyásol negatívan más, pozitívabban gondolkodó tagállamokat.
Amikor a médiában GM téma kerül terítékre, akkor általában...
When GM is discussed in the media, the talk is usually about production and environmental protection. But little is said about the benefit to the consumer in the form of cheaper foodstuffs. It is also to the benefit of the environment if fewer chemicals are sprayed, together with savings on chemicals and energy. The media should concentrate on the economic advantages of GM crops.
Erőforrás megtakarítással is jár.
A média felelőssége mindenképpen óriási.
The media have enormous responsibility. A European survey showed that people obtain most of their scientific knowledge from the media. So what we write is extremely important in forming opinions. What people buy in the shops is influenced by what they see and hear in the news, rather than by adverts, especially in Hungary. So we must not be taken in by either side, but must obtain correct information. In the EU, the GMO debate is a political matter, where business interests may influence decisions made in the European Parliament. This has repercussions on Hungarian politics and on the Hungarian press. In the 1970s, the scientists themselves asked for a moratorium when the first gene technologies appeared, but this was soon suspended. Unless there are real scientific counter-arguments, the advance of science cannot be held up for long. After 30 years in science journalism, I feel that scientific development overrides every type of political or media interest.
Sok megjelentek, maguk a kutatók vállaltak egy önkéntes moratóriumot, aminek aztán a hatása sem volt olyan hosszú ideig tartó, és aztán el is múlt a dolog.
The theory of evolution, published by Charles Darwin, and the conclusions drawn by Gregor Mendel in his experiments on peas were also disputed in their day. Many years passed before these results were accepted, either in scientific circles or by the general public. However, theories once considered to be shocking are now taught in schools to children who carry in their genes the thirst for knowledge and for further development.